But I am going to talk about the question of what regulates cell size in phytoplankton communities. So this is a new topic for me. I'm pretty excited about this. I think it has relevance to the HAB problem. So let me give you a little bit of an introduction. My research is part of a long-term program of ecosystem research in San Francisco Bay that's run by the U.S. Geological Survey. And the goals of this research are to understand how estuarine coastal ecosystems respond to human drivers of variability, things like species introductions, water diversions, river damming, habitat, landscape transformations, habitat restoration, and also how these ecosystems respond to climate variability at scales from events like heat waves to multi-decadal climate oscillations. And within our group, the biological community that we focus on is the phytoplankton community. And beginning maybe a year or so ago, Rafe Cadella started placing his one of his imaging flow cytobots on our research vessel. And so we're continually taking images of phytoplankton as we sample along the estuarine salinity gradient of San Francisco Bay. And as you know, this is a spectacular new technology that allows us to view phytoplankton communities at levels that we could never visualize them before. So I'm showing here 4 different images that were taken at about the same geographic location in San Francisco Bay, but on different dates. And when you look at these images, a number of things stand out. The first thing that stands out to me is just the diversity of forms. And of course, these samples represent a minuscule part of the real diversity of the phytoplankton. The second thing that stands out, and it's kind of the question that I want to talk about, is how variable the community makeup is from time to time. I mean, we don't need to do any kind of analysis of these images to see that the communities were made up of different shapes and forms and sizes at different times. So the phytoplankton community makeup is highly dynamic. It changes over time. And it can change rapidly over timescales of days or weeks. And in some ways, this is the fundamental ecological question of phytoplankton dynamics. What are the underlying mechanisms that give rise to this kind of community variability? And another feature of the problem that emerges from looking at these images is that these images depict 2 very different levels at which we have assessed phytoplankton communities. The first is on the basis of species assemblages, thinking of communities as assemblages of species based on morphology. And so many of you, I think, can recognize some of the morphologies and species present in these samples in San Francisco Bay. And as essential as this problem is to understand community dynamics at the level of species, we've made what I would judge to be remarkably little progress on this really challenging problem, even though we've been working on it at some level for probably more than a century. We just don't have the knowledge, because of the complexity of the problem, we just don't have the knowledge or the skill or capability to understand and explain, model, hindcast, or forecast changes in phytoplankton communities at the level of individual species. This remains a really challenging problem. The other level that we can visualize of community assembly here is on the basis of cell size. And this is a different story. So you can see part of the spectrum of phytoplankton cell size that ranges by a factor of 10 to the 9. This represents a small part of that spectrum. But when we start looking at phytoplankton communities organized on the basis of size structure, we're making important rapid advances. And I think we have much more success at understanding the dynamics of communities when we simplify the problem by binning cells into size classes. And I think the reason for this, the fundamental reason for this, is that organisms and their population dynamics are constrained by fundamental biophysical rules. And we've known this since the beginning of ecology, and we're thinking about work over a century ago by the population biologist Elton, who conceived some fundamental scaling rules of community assembly. So biological communities are organized by size. Small organisms are abundant, large organisms are rare. Small organisms operate fast. For example, they reproduce more quickly than large organisms, and larger organisms feed on smaller. So these were basic concepts of size-based rules that were built from his work of terrestrial ecosystems, but they apply equally well to pelagic ecology, to plankton communities. And I think that the fact that organisms are constrained, their growth rates, their resource acquisition rates, their predation rates are constrained at some level by the biophysics associated with size. I think this is the fundamental reason why we're making better progress at understanding community dynamics on the basis of size rather than species. Okay, so I've organized this talk into 4 parts, and the first part I want to talk about what we've learned about phytoplankton size distributions from our multi-decadal study of San Francisco Bay. So this map shows the distribution of sampling sites where we collect water samples and analyze them by microscopy. So this is standard light microscopy results that I want to talk about. So from 1992 through this year, we've collected 1,214 samples that were analyzed by light microscopy. And in the process, almost 25,000 individual cells were counted and measured. So this is a pretty robust data set for looking at size distributions. And from this data set, 3 size-based patterns emerge. So let me explain this figure. The x-axis here represents the biovolume of phytoplankton cells in a sample. So it's cubic microns of cell biovolume per milliliter. And the x-axis represents, and each dot here represents an individual sample. These are the 1,214 samples. And the y-axis represents cell size. And in this case, we've taken the volumetric cell size, converted into a linear dimension, equivalent spherical diameter, and this is on a log scale. After all those words, the pattern that emerges here is that as the total cell biovolume in a sample goes up, so does the mean cell size. So as the community builds, it builds towards large cells. I think this is a really fascinating pattern. It's not unique to San Francisco Bay. It's been seen in other places. So this seems to be a common size-based pattern that as biomass builds, cell size goes up. One of the steps that I did to try to simplify this problem is look at the size distribution of all of these 25,000 cell measurements and bin them into 5 different size classes. So these are the quintiles of the distribution. This is again on a log scale. This is a log transformation of cell size, and then looking at the distribution, breaking it up into quintiles of equal numbers of observations. And then I calculated the mean cell size in each of these bins. So the smallest, there we go, I just did it. The smallest size class and then I calculated the mean cell size in the smallest size class. It's 3.5 microns, that linear dimension. The largest size class, the mean cell size is 39 microns. And so we bin all of these cell measurements into these 5 size classes. And then a second pattern emerges where I ask the question, if we take the biovolume from all of the samples, sum it up, and partition it into these 5 size classes, how do the contributions of the different 5 classes fall out? And what you see here is that the largest size class contributes the vast majority of the total biovolume in all of those samples. And this is what I mean when I say that large cells rule in estuaries. They contribute 69% of the cumulative biovolume in all the samples that we've collected. And the 4 smaller size classes contributed each less than about 10% of the biomass. So in San Francisco Bay, large cells rule in terms of their contribution to overall biovolume. And then the third pattern is that phytoplankton blooms in this estuary are the result of selective biomass growth of the largest size class. So this is a plot where the y-axis is the amplification factor of biomass during blooms for each of the 5 size classes. So you know, we don't have a uniform definition of what a bloom is. I just arbitrarily said, let's look at the samples that are in the top 10th percentile in terms of total phytoplankton biovolume, call those bloom samples and the other 90% non-bloom samples, and then for each size class, calculate the mean ratio of biomass during blooms compared to non-blooms. And you can see that for the largest size class, their biovolume was amplified by a factor of 60, so a 60-fold increase in the biovolume of large cells during blooms compared to non-blooms. But for the other 4 smaller size classes, their biovolume didn't change much during blooms. So blooms are selective growth of large cells in this estuary. And those large cells are primarily diatoms, large diatoms, and secondarily dinoflagellates. And among the collection of large cells are several taxa that are hab-forming taxa in other estuaries. And Morgane McGibbon has a poster on this today in T20 or T20. And then later this morning, Misty Peacock, so Morgane's poster is about these taxa, these hab-forming taxa, and their abundances in the bay. And later this morning, Misty Peacock is going to give a fast talk about algal toxins in San Francisco Bay. Okay, now I know that many of you are asking, well, you know, what about the picoplankton that aren't captured in and aren't observed by traditional light microscopy? So we've tried to address this question a couple of different ways. One is by looking at enumerating small cells, picocyanobacteria, with epifluorescent microscopy, and the basic results of that is that those organisms are, of course, always there, but they're not major contributors to phytoplankton biomass in the system. And these are results showing the relative contributions of the different algal groups on the basis of their pigment, their diagnostic pigments. Each of these bar graphs represents a different geographic region in San Francisco Bay. These are the results of analyses of almost 500 samples for pigments that are analyzed by Chemtax by Misty Peacock when she was a postdoc at Santa Cruz with Rafe Kudela. The bottom line is that it gives a very similar result to the light microscopy. That is that 65% of the pigment concentrations were, chlorophyll concentration was contributed by diatoms, 14% by dinoflagellates, and only 4% by cyanobacteria. The point of this is that light microscopy has a bias by not detecting, counting, and including the biomass contributions of the picoplankton, but these additional methods give us confidence that the errors that we're making in the partitioning of biomass into size classes are not large. Okay, and the last thing that I want to say in this part of the talk is that this result of dominance of biomass, and in particular biomass during blooms by large cells, by diatoms and dinoflagellates, is not unique to San Francisco Bay. So this is a paper that we published a couple of years ago in which we compiled phytoplankton data sets that were analyzed similarly by light microscopy from 86 different sites. And these are well-studied sites, and many of them are around the estuary out the back door here, Chesapeake Bay. Also, a large number of sites around along the Danish coastline. Some sites in the Baltic, some sites in the Neuse River where Hans Perl works, and 4 of our sites in San Francisco Bay. And the bottom line is that, in this case, cell volumes were converted into carbon biomass using the standard conversion equations. And the net result of this analysis is that for these 86 coastal sites, these are high latitude or temperate zone sites that are at the land-sea interface. Nutrient concentrations at most are pretty high. So for these kinds of environments, diatoms made up 58% of the carbon biomass and dinoflagellates 19% of the. So this rule that large cells are dominant in these kinds of estuarine coastal habitats seems to be a general rule. It's not specific and unique to San Francisco Bay. So the question is why? Why is that? What is it about these places that selects for large cells and diatoms and dinoflagellates in particular. And the way in which I tried to answer this question is with a model. So I want to describe for you a size-structured model and then show you how the model was built and the kinds of experiments that I did with the model to try to get at this question of why large cells rule. And I was able to put this model together because of this really fantastic paper that was published in 2014 by Darcy Taniguchi and her co-authors at Scripps. And so this is an NPZ model. It's a nitrogen phytoplankton zooplankton model, but there are 5 size classes of phytoplankton, 5 corresponding sizes of zooplankton in one nutrient pool, the pool of dissolved inorganic nitrogen. So I basically took this model that Darcy Taniguchi produced. I think this was part of her PhD dissertation. And I tailored it to the environment of San Francisco Bay to see if we can use model simulations to get at this question of why large cells rule. And this model is a set of 11 ordinary differential equations, 1 for nitrogen, 5 for the 5 phytoplankton size classes, 5 for the 5 zooplankton size classes. The zooplankton size classes were chosen as the largest cell size in the corresponding phytoplankton size class. So the smallest grazers feed only on the smallest producers. And those equations are structured like this. So the time rate of change of, it is so easy to hit the wrong button on this. The time rate of change of a phytoplankton biomass is biomass times the net growth rate, where the net growth rate is the cell division rate, the mean cell division rate in that size class, minus the specific grazing rate. And then in the model, growth rate is calculated as a function of phytoplankton size. That's the whole point of this model, to understand what size-based rules of growth and grazing mean, temperature, and then really simple representations of regulation of cell division rate by light and nutrients. And then for zooplankton grazing, the grazing rate, again, is a function of size of the grazers, temperature, and phytoplankton biomass. And embedded in the model are size-based scaling rules that actually trace back to the work of Elton. This is something that we've known for a long time, that biological rates follow power functions of size. And so the maximum phytoplankton growth rate is a power function of cell size, expressed as this linear metric of equivalent spherical diameter. And similarly, the maximum zooplankton grazing rate is calculated as another power function. And so in order to build and run a model like this, we need to know the coefficients of these 2 power functions. And one of the reasons why I think this paper of Darcy Taniguchi is so important is that she has scoured the literature. I think she's done the most comprehensive compilation of all of the experimental measurements of grazing rates and growth rates as a function of cell size. And in her paper is this little but incredibly valuable table which gives the coefficients for these power functions. And these coefficients are based on hundreds of laboratory experiments by many different laboratories. So for example, in this table are the coefficients. This is the constant and the exponent of that power function that describes phytoplankton maximum growth rate as a function of cell size. And the other parameters that we need in this model are also mean results of many different kinds of experiments. In the paper, in the supplementary information of the paper, is a listing of all of the different measurements that have been made in laboratory experiments, the organism, the cell size of that organism, the temperature, and the rate of the process that was measured in those experiments. So this compilation it builds from others that other people have done, and in itself is really valuable. So these parameters were embedded in the size-structured model, and so we have we now have these mean size-based rules that we can use to start modeling phytoplankton size-structure dynamics. So this is what one of the size-based rules looks like. I'm sorry I'm going on the right. I can't do both at the same time. So this is what the size-based rule looks like for the maximum growth rate. So this is maximum growth rate is a function of cell size. This is that power function. The blue dots here, circles, represent the mean cell size for the 5 size classes that emerged from analyzing the phytoplankton data set. And so you can see that small is fast and large is slow. And now we know this in a quantitative way, at least in the mean. The other thing that Darcy did in this paper was take the measurements of rates across different temperatures and calculate the mean Q10s for these processes. So the mean Q10 for phytoplankton growth rate is 1.62. And so this is what that power function looks like at a temperature of 10 degrees instead of 20. The main point of this now is that we have scaling rules that are grounded in this rich history of experimental measurements that allow us to measure, to estimate the maximum phytoplankton growth rate if we know cell size and temperature. And the same kind of scaling rule for grazing rate. So she did a similar kind of analysis of grazing rate studies as a function of grazer size. You can see that this power function has a larger exponent. It decreases more quickly. And again, the blue circles here represent the 5 size classes of grazers in this model that I'm going to show you results of soon. Another important and interesting result from this study is that the experimental observations tell us that the Q10 for grazing is larger than the Q10 for phytoplankton growth rate. And it turns out this is really important, and I'm going to show you why. And we have size-based rules for other things, like the half saturation constant for nitrogen uptake. And we've known for a long time that small cells have smaller half saturation constants. They're more efficient at acquiring nutrients in oligotrophic states. But now we have an equation that gives us the mean half saturation constant as a function of cell size based again on all of these different experimental measurements. Okay, so with that as background, I used this model with all of these size-based rules to simulate changes in phytoplankton and zooplankton biomass using environmental measurements from San Francisco Bay. So I ran the model for a 3-year period, and the forcings of the model in the upper left here are measurements of temperature. We make measurements every month or twice a month, so I interpolated to get daily values. So the model is doing computations daily. So we have 3 years of this temperature cycle, and then next to that, light. And light is simply the depth integrated mean photosynthetically available radiation over the height of the water column. And I specified the water column as a 10-meter water column. So the model was forced with data from San Francisco Bay. All of the parameters are from this compilation of the Taniguchi paper. And the model results are shown in the bottom here. So the bottom 3 panels show the time series of simulated biomass of the smallest phytoplankton and zooplankton component of the system. So the red lines are the simulated biomass of phytoplankton. The green lines, I'm not even sure if you can see them, are the simulated biomass of zooplankton. This panel in the middle is for the intermediate size class, and the panel to the right is for the large cells. And one thing that stands out pretty clearly here, I should point out that the y-axis here is a log scale. So for the smallest size classes of phytoplankton, if you look at the time series of simulated biomass, they're characterized by this high frequency but low amplitude variability. So they're really not doing much. They're oscillating around a small biomass. There are these high frequency variations. And that's very different from what the model tells us the large cells would do in a system like this. So if we look at the simulations in the lower right panel, the red line is simulated biomass of the large cells, and again, this is on a log scale. And what you see is that blooms develop in the model, And the blooms develop and peak during the springtime. They're recurrent, but they vary in magnitude from year to year. And as those blooms peak, they draw down the pool of dissolved inorganic nitrogen to levels that are near detection. And this is a pretty characteristic feature of San Francisco Bay. Spring blooms of large cells that during some years draw down the pool of dissolved inorganic nitrogen to very low levels. But the point is that the large cells behave in this model very different than the small cells. And that behavioral difference has in its roots differences in size-based rates of growth and grazing, as I'm going to show you. One of the things I did with the model output was I treated each day of simulation as though it was a measurement in the bay, and I treated that as a data set and analyzed it the same way I analyzed the measurements from San Francisco Bay. And the model results reproduce these three size-based patterns that I showed you. So on the left panel here, the x-axis is phytoplankton biomass. Each dot is a day in the 3 years of simulation. And then the y-axis here is mean cell size. So the same pattern in the model that we saw in the data, as biomass goes up, cell size goes up. We're using the exact same size classes in the model as we are in the data. And if we partition the biomass that's produced in the model over the 3-year period into the 5 size classes, most of the biomass is produced by the large cells. So large cells rule in the model like they do in the data. And then the same thing is, the reason for that is that these blooms are largely selective biomass growth of the largest size class. So the fact that the model reproduces these fundamental size-based patterns gives me at least a little bit of confidence that we can keep playing with this model. And one of the ways that we can play with the model is to examine rates that are calculated in the model and the distribution of those rates by cell size. So the upper panel here these box plots show the distributions of the calculated growth rate of each of the 5 size classes going from smallest to largest. And I think you can see this trend that the smallest cells have a higher mean growth rate than the largest cells. And of course, that's embedded in the model. That's that power function that we calculate maximum growth rate from. The middle panel here shows the distribution of calculated values for the net growth rate. And the net growth rate is what's really important, because that's the growth rate that drives population variability. And if you look at the distributions here, there is no difference in the mean net growth rate of the different size classes. And the reason for that is that even though small cells divide faster than large cells, small cells are also consumed by faster grazing consumers than the large cells are. And the net result of this is that there's no difference in the mean net growth rate across the 5 size classes. And then lastly, if we look at daily production, which is just the net growth rate times the biomass, the 4 smallest size classes don't have much variability of daily production, whereas the large cells have these boom-bust cycles. And the booms are these periods of explosive growth of biomass as blooms grow, and the busts are these periods of equally rapid biomass production decline as blooms decay. So this leaves a paradox. It doesn't explain why large cells are selectively growing during blooms. It doesn't explain why large cells are the dominant component of biomass in the mean, because their net growth rates aren't any different on the average than the net growth rates of small cells. So the mean of the net growth doesn't seem to be very important. But what does seem to be important is the pattern of variability of the net growth rate. And so that's what I want to show next. So you've seen this panel before. This is the panel showing the simulation results of biomass for the 3 components. And the bottom panel here, these green lines here, plot the time series of days when the net growth rate was positive. And I'm not showing the dates when the net growth rate was negative. And if you look at the patterns of variability for the smaller size classes and compare them to the large size class, they have a really different pattern. And that different pattern is that, again, for the small size classes, there are these high frequency oscillations between positive and and negative net growth rate. But for the large cells, there are these runs, these long periods of sustained positive net growth rate. So it looks like the duration of these runs of positive net growth rate are the explanation of why blooms can develop. So even though the net growth rate might not be very large, it might not be a large rate, if it's sustained over a long enough period of time in a continuous series, that allows biomass to grow exponentially and build to these high levels that we see during the springtime. So it looks like the difference between large and small cells is these runs of sustained positive net growth rate that are a result of the fact that the large grazers graze more slowly, their populations grow more slowly than the small grazers, and that leads to lags in the population response of the grazers that is present in the large component but not on the small. And this is not a new idea, but I think I don't remember seeing this representation of the idea in a model before. The basic message here, the basic lesson here, is that large cells can gain an advantage under conditions when we can divide rapidly because there are lags in the response of their grazers. So the variability of the net growth rate is an important part of the size dynamics. Okay, so this brings us again back to Elton and the concept of size-based rules. And this is a plot again showing the maximum grazing rate as a function of size. And it's just a reminder that small grazers can feed rapidly and divide rapidly, so there's tight coupling between small and large, between small producers and consumers, whereas the large grazers are grazing at a slower rate, their production is slower, their turnover rates are slower, and that creates these windows of opportunity for large cell biomass to grow. So, you know, this rule that small is fast and large is slow is at the root of the explanation of why large cells rule in this model. Now, of course, temperature is also really important. Cell size doesn't explain everything. And one of the things that came out of the model is that if you plot biomass of the large cell component, so I'm going to focus a bit now on the large cells. If you plot a biomass of the large cell component as a function of temperature, you get this really strange distribution, and it shows that the large cells didn't have biomass at low temperatures or at high temperatures, but they peaked at temperatures around 15 degrees centigrade. And I was kind of struck by the comment that Don Anderson made yesterday about Alexandrium not developing blooms after temperatures of about 15 degrees. I don't know if there's a connection between this and that comment or not, but the explanation for this, the explanation of why blooms developed at temperatures around 15 degrees is explained by, again, this temperature-based scaling rule. And this bottom graph is showing what I'm calling the maximum net growth rate. It's just, and this is, again, is of the large cells. So it's the difference between the maximum growth rate of large cells as a function of size and temperature minus the maximum grazing rate. And what you see is that at low temperatures, the maximum net growth rate is a positive value. And at about 15 degrees, it's about 0. And then after 15 degrees, it decreases very rapidly. And this is an expression of the fact that phytoplankton growth rate has a smaller Q10 than zooplankton grazing rate. So as seasonal temperatures warm, grazing rate accelerates faster than growth rate. Okay. Next, I use this model to do some really simple-minded experiments, but I think they can generate some hypotheses that we can test. So the first experiment that I did was I used the model results that I've just shown you as a baseline, and then I asked the question, what happens if we just turn grazing off? What happens if we set grazing to 0 and run the model again with the exact same initial conditions and forcings? And the result, and it's not surprising, the result is very different. So the left-hand panel is the baseline simulation showing that large cells rule when grazing is present. The bar graphs on the right-hand side show the size distribution of phytoplankton biomass produced over this 3-year simulation binned into the 5 size classes, and it's the inverse pattern. So here now, small cells dominate biomass. The largest cells are the smallest component. And I think even though this is a very simple-minded experiment, I think it illustrates pretty clearly a very important point, and that is that grazing can structure the size distribution of phytoplankton communities. And if it can structure the size distribution, it probably plays an important role in shaping what species are present as well. So in this case, small cells just win because they grow faster than large cells if we turn the grazing off. As I said, San Francisco Bay is an example of a high nutrient estuary. And so the next experiment that I did was to address the question, what would happen if we transformed this estuary into an oligotrophic system? So instead of the initial condition of 48 micromolar dissolved inorganic nitrogen, which is the long-term mean for January, I set it to 1. So turn San Francisco Bay into an oligotrophic system. And in this case, large cells don't win, they lose. And the reason why they lose is because of this scaling relationship between size and the half saturation constant. So they're inefficient, and this is something again that we've known for a long time. They're inefficient at acquiring nutrients in low nutrient environments because of their large cell size. So there's a more even distribution of cell sizes, and the large cells lose in the oligotrophic simulation. And then the last question that I addressed with a similar kind of model experiment is, I'm really curious about this 15 degrees threshold and if it's real and if it's important. So I asked the question, what would happen if we take San Francisco Bay, which is at 38 degrees north, and move it towards the equator and add increased temperature every day by 5 degrees centigrade? So this is a warming simulation. And in this case, the large cell blooms largely disappear in this simulation. And the reason for that is that we've elevated the grazing rate. And so these windows of opportunity for large cells for their biomass to grow exponentially become diminished as the estuary is warmed. So if we take these 3 experiments together and put them together in a framework, this is what one kind of framework might look like. And it answers the question, where along combinations of gradients of temperature and nutrients would we expect large cells to rule? Rule in terms of developing high biomass blooms. And so if the world of plankton is run solely on the basis of size-based rules, this is a framework that emerges from those models. And the framework says that large cells can rule only in high nutrient, cool conditions. And when we get into warmer conditions, they can't rule because the large grazer, the rate of grazing by the large grazers is accelerating in a warming climate. And in low nutrient environments, small cells win because of their efficiency at acquiring nutrients in low nutrient environments. And if the world was run solely on the basis of size-based rules, if this framework is an accurate representation of temperature and nutrient combinations that would select for large cells or small cells, a couple of hypotheses emerge, and these are hypotheses that I think are testable. So one hypothesis is that as we increase nutrient concentrations in water bodies, there's a progressive tendency to select for the growth of large cells. And so, of course, this has relevance to a problem that is at the core of science in Chesapeake Bay. That is the problem of what have been the effects of nutrient enrichment. So this is a graph from a paper of Walter Boynton's in which he has reconstructed the loading of both nitrogen and phosphorus into Chesapeake Bay going back to the 17th century. And it's now well established that consequences of this nutrient enrichment have been increases in phytoplankton biomass, increases in productivity, increased metabolism of the estuary leading to hypoxia bottom waters when the estuary is stratified during the summertime. I'm not sure how much we've talked about or asked the question of what the role of nutrient enrichment might be in terms of changing the size distribution of phytoplankton cells. And the model simulations, experiments that I've done here suggest that another consequence of anthropogenic nutrient enrichment could be increasing cell size. And if that increased cell size is increasing diatom component, that exacerbates the problem, because not only are we producing more, we're producing more in forms that sink to the bottom where they're metabolized. I don't know how to test this hypothesis, and I wonder if it's possible to use sediment cores to try to reconstruct sizes of diatoms in the sediments during this era of nutrient enrichment. The second hypothesis that occurred to me is that large cell blooms can only develop when temperature is below some critical value, and the model parameters that I've used here suggest that it's around 15 degrees centigrade. This is a hypothesis that I think would be pretty easy to test in a series of mesocosm experiments in which we take plankton communities, include all size classes, look at outcomes when we modify temperature that spans this 15 degree threshold. A third hypothesis, again a hypothesis that is testable, is that cell size diminishes along temperature gradients from high to low latitudes. I'm not aware of any compilation of phytoplankton and temperature data sets along coastlines that span from the boreal to temperate to subtropical to tropical latitudes and temperatures. That's a measurement program that is feasible, that could be done, and it would be an opportunity for us to test this hypothesis that there are latitudinal gradients in the relative importance of large versus small cells. And then following this one, the hypothesis that large cell blooms will disappear if temperature increases beyond a critical threshold. So we are imagining and anticipating expansion. It is so easy to do that. Expansion of these latitudinal bands of temperature towards the poles, and places that in today's climate have seasonal temperatures that fall below 15 degrees centigrade in the future might not. So the fourth hypothesis is that if that happens, we could anticipate changes in cell size, and in particular loss of the large cell component. Okay, so this is the last part of this presentation, and this talk has been a lot about rules. It's about size-based rules and temperature-based rules, and rules of when large cells can dominate along gradients of temperature and nutrients. And I just want to remind you that this pattern, these rules, are only true if it's the size-based traits of phytoplankton and zooplankton that determine community composition. So one of the things one of the attributes of this model is that there is no way that this particular model that's based only on size-based rules can generate blooms of small cells. And I think that many of you know examples of, and probably many of you are working in places that do have blooms of small cells. And so these are what I'm calling the rule breakers. And just kind of off the top of my head when I was putting this talk together, I thought about 3 papers that I knew of. And I'm going to ask you for more examples. So here's one example. This is an example that you probably all know about, this really remarkable, I think it was an 8-year continuous bloom of a pelagophyte in Laguna Madre in Texas. And so this pelagophyte broke the rule, and the rule is tight coupling between small producers and small grazers so that the small producers don't have opportunities for their biomass to build. That's what the size-based rule tells us. But this pelagophyte has apparently evolved an adaptation that allows it to break that rule by escaping the constraints of size. And the author, Ed Buskey, and other authors of this explained this on the basis of a couple of traits of this pelagophyte. One is that it is adapted to hypersalinity, I think a key to the development of these blooms was sustained drought leading to hypersalinity of the lagoon. And this particular organism was adapted to those hypersalinities. The small grazers were not. And also production of a chemical inhibitor of grazers. So here's an example of a small organism that produced a harmful algal bloom as a result of traits that it has that are unrelated to cell size that allow it to break away from the constraints imposed by size. Another example that I remember when this event happened, I think many of you do too, this fish-killing bloom of Chrysochromulina along the Scandinavian coast in 1988. Many papers were written on this because of its importance. And the authors of this paper explained that Chrysochromulina could develop high biomass blooms as a result of the fact that, first of all, it's got multiple trophic modes. It's not just autotrophic. And the model that I had no other modes of nutrition other than photosynthesis. And it produces a toxin that inhibits grazers and other algae. Again, these are traits that allowed for this organism to escape the constraints of size-based rules and develop blooms. And the third example that I'll give you is a really nice paper by Ed Flips and his colleagues who are working in lagoons of Florida. This paper is about red tides, green, and brown tides. And I just want to focus on the green tides, which were high biomass blooms of pico eukaryotes and cyanobacteria. And the explanation for the ability of these organisms to escape, to break the rules, was that these are organisms, these are algal forms and cyanobacterial forms that have the capability of accessing forms of nutrients other than inorganic forms, and also a climate anomaly that kind of set the stage for this. A cold anomaly that apparently suppressed the small grazers for a long enough period of time for these small cells to develop blooms. Okay, so to kind of wrap this up, based on work that ecologists were doing a century ago and building from that work, the theories and experiments, I think we're at the point now where we have developed size-based rules of how phytoplankton communities are assembled on the basis of phytoplankton cell size and grazer size. Secondly, these are grounded in strong, both theoretical and empirical. They have a strong empirical and theoretical foundation. That from these rules emerge patterns and hypotheses, but not all species follow the rules, and other traits besides size-based traits are important in terms of understanding community variability. So with this as kind of a wrap-up of the importance of size and the fact that it sets rules, but there are many examples of rule breakers, I'm going to end this with two questions that I would like to ask you. I don't know the answer to either of these questions, but the body of knowledge contained in this room, I think, has a lot to contribute to answers to these questions. So I'm going to end with questions rather than answering questions, and I'd like to hear from you. And the first is, are all taxa rule breakers? Are all of the organisms that you all are working on, do they have traits unrelated to size that allow them to break the constraints, and in particular the constraints of grazing that allow their biomass to build that otherwise wouldn't happen if the size-based rules were the only ones that were operating? And when I'm talking about rule breakers, in particular, I'm thinking about other examples of places where small cells develop high biomass blooms, because the size-based rules tell us that small cells shouldn't be able to acquire a high biomass because of this tight coupling between their production and their grazing by fast-growing zooplankton. And in addition to that, rule breakers would be occurrences of high biomass large cell blooms in temperatures greater than about 15 degrees. So I'd be curious of examples of high biomass large cell blooms that develop where temperature is greater than, say, 20 degrees. Off the top of my head, I don't know of examples, but I suspect that you do have examples. And then the last question that I want to leave you with, and I hope you say something in response to, are what are the traits that allow these habitats to break the rules? And we heard a lot about this yesterday. It was kind of a theme of the talks yesterday. I think it's a theme that's going to be prominent throughout this session. And so traits that allow phytoplankton species to break the size-based rules include things like buoyancy regulation and motility. I didn't build anything like that into the model that I described. Multiple mechanisms of escaping or deterring grazers. I talked a little bit about that in those 3 examples. Colony formation is a way of deterring predators. We heard something about allelopathy yesterday. I know very little about this. I'd like to learn more from you. Mixotrophy, there were no other modes of nutrition in the model. I'd like to know what the role of mixotrophy is in terms of supporting blooms of species that form harmful algal blooms. Life cycles, we heard a little bit yesterday about cysts and cyst cycles. The ability of organisms to take up organic nutrients as an advantage that also was not built into this model. So I'm hoping to use whatever time is left here to learn from you and hear your thoughts and experiences on the first question of whether HABs are all rule breakers, and the second question of what is it that allows them to break the rules. So thank you very much.
Can I start? So Carlo is a breaker, and it's because Carlodinium veneficum, 10 micron, but it has the 2 modes, mixotrophy, anti-grazing compound that it makes. So that whole class, and it will not bloom. And this is in the bay with the big guys like Akashiwo and so forth you've shown. Now the other side of that is Noctiluca, which is the big-ass cell, which fits your rule really, really well.
Yeah, but that's not an autotroph.
Correct.
Yeah. So what kind of chlorophyll, when you say have Carlodinium,
Yeah.
when you observe Carlodinium blooms, I'm curious, what kind of biomass, what kind of chlorophyll levels can they develop to?
Cell densities, depending locally, can actually get up to 800,000 cells per mil.
Okay, this is an absolute rule breaker then.
Yeah.
And the attribution is mixotrophy and...
So it goes from being a really poor phototroph, dividing every 4 days. When it's eating, it divides every day. Its food items are these things called cryptophytes, which are out there everywhere, basically.
Yeah. So it's escaping the rules by breaking away from being an autotroph then.
But then it also, this compound that it makes, which it uses to capture the prey, is also anti-grazing.
Okay, great example. Yeah.
Come on, I'm sure there's others out there.
Great talk. I have Clarissa Anderson, by the way. It's nice to finally see you and meet you in person. I have a question. I'm thinking about the net growth rates that you saw that were at this sort of mean, steady state, despite these pulses of positive net growth rate that you then delved into. And I'm thinking about this in the context of the other sort of suggested paradox in the bay, which is potential ammonium inhibition of nitrate uptake. And what would it mean to go back and say, take a time series of satellite imagery and actually extract particle size distribution and look at that relative to nutrient loading in the bay? Because I think that the reason that these hypotheses still exist is because that net growth rate that you saw is masking the nitrate uptake over time.
It would be really easy to add that process to the model. And do those kind of modeling experiments. I don't know what kinds of historical data are available to test this idea.
Well, there's nutrient time series and then there's ocean color time series. So I think the 2 of those could be matched.
So can ocean color time series work in a turbid estuary where the photic depth is
Well,
like
there have been advancements with the hyperspectral imagery. Of course, there's not a lot of that.
Yeah. It's an interesting idea. It's one that I hadn't thought about. I haven't thought much about using satellite imagery in San Francisco Bay because of its high turbidity, but it would be an interesting idea to explore.
Okay.
Thanks.
Coming, Carm.
You got another one?
Good morning, Jim. I'm over here, towards the middle.
Oh, okay, okay.
Two things. One, I think that excellent talk. I really enjoyed it. There's one element here that I think is may help explain a lot of the exceptions or the rule breakers, if you will, and that's the concept of turbulence, which is not in this at all.
Yeah.
And of course, you know, that goes back to Margalef and then later on Smader and Reynolds and so forth. So that there's another element in there that I think has to be in consideration. And that may be one of the physical properties that allows some of these species to do things that we normally wouldn't fit in terms of just plain growth and size. The second thing I want to comment with is that the 15-degree thermal window, if you want to call it that, or thermal niche as it's been called, is been repeated over and over and over in estuaries, particularly in Narragansett Bay, for instance, where we've had data going back from the 50s all the way up to the 90s in species composition and changes with time. And at the Heterosigma cascio blooms, for instance, there, 17 years worth, clustered around that 15-degree threshold all the time. So that's one worth looking at again, and it's a fairly small species.
What was the explanation for that?
Again, it's a change over time. It's basically a time when diatom blooms are collapsing, and there's a transition into a spring-summer period. Also, there's a variability in the concentration of zooplankton and the most dominant species at that time. So, and in fact, it's based on the audicology of the organism, that 15 to 17 degree range is where you get the maximum growth rates.
Okay, but so there wasn't consideration given to this difference in the Q10s of grazing and growth rate then?
Yes, there is. But I think in this instance, it's the net change that's happening in this mixed community, again, with grazers, with other species that they're competing against in terms of nutrients.
Okay, I need to go back and reread these papers. Your point about turbulence is of course, something that we absolutely have to consider, and it's one of the reasons why I listed motility and buoyancy regulation here as one of the non-size-based traits. I mean, the importance of motility and buoyancy regulation is in the context of ways to overcome mixing from turbulence. It gives species advantages when they can accumulate on the surface, for example. So I think, it would be a relatively straightforward next step to put this zero-dimensional model in a spatial framework and start with a vertical 1D model where you can look at different scenarios of turbulence and add these behaviors of motility and buoyancy regulation. I think it's something that has to be done. I think this is, the model that I've done is the simplest that you could imagine, and some ideas and hypotheses emerge from it. It's far from real, but I think there are steps that we can take to add realism to the model, and incorporating the importance of turbulence and the traits that phytoplankton have to adapt to variable turbulence is something that needs to be done.
Okay, thank you.
Coming over. You got it? While that's going on, this 15-degree, it's all driven basically by the temperature dependence of the grazing, right?
That's right. Well, the balance between
No, correct. I understand that. So, but that's an evolvable feature. At some point, maybe that could switch, that curve not being so steep. The grazers could, in fact, with time, with climate change, maybe?
So I think a really important point to remember here is that those scaling rules, the Q10 rule, for example, is the mean of many different experiments, and there's variability around that mean. So another question that I've got is how do we build in that variability around the mean, and what does that mean? And what patterns emerge if we include that variability around the mean?
Yeah.
Hi there, Abir Banerjee, EPA. This is a very minor component compared to what's up on the screen right now, but I thought I'd mention something that crossed my mind, which may be important in certain contexts. symbiotic associations are a big thing in some cases. We learned a little bit yesterday about when these harmful algae or stuff are associated with particular hosts. But I can think of a few examples where having a symbiotic association might allow them to gain mobility or to have refuge space for disturbances, and escape from predation, escape from physical agitation. My brain stopped working. But you get the point I'm making.
Yeah. So is there a specific example that you could send me, a paper that I could learn from?
I'm going to have to double check it, but what crossed my mind, oddly enough, was a case of some toxic cyanobacteria associating with Vorticella, a protist which has a little more mobility. There are other examples which are a little more obvious, things like associations with macroalgae and some aquatic submerged plants, which will provide shelter from predators and sort of provide a base to prevent turbidity and stuff from affecting them.
Send me examples, I'd appreciate it.
Will do.
Okay. Thank you very much.
You're going to be the last, okay?